Thank you, Caroline, and thank you for joining me this afternoon here in Singapore. I don't know what time of the day is and where you may be, but I'm going to tell you a little bit about some stories associated with research in our group. And our group is mainly a chemistry-based group. We like to make new molecules, and we like to think about how do we apply those molecules for enabling a particular technology and what that technology might do in the future. So even if you are not in chemistry, you might want to try to sort of pay attention to how developing skills in a particular area of science and engineering is a very useful tool to being able to be influential in impacting society in many different ways. So I started my career as a chemist. At your time, when I was trying to decide the graduate school, I had decided I wanted to be a chemist. I really like to make molecules. I think molecules are very interesting. I like to think, how do you put them together and what they're good for? So synthesis is by definition the way that we make new molecules. I also became interested in materials. Materials, there's usually a performance that you're interested in maximizing because ultimately there's a technology that you're trying to impact. And people like technology because, well, they can generate wealth. They're relevant for national defense. They're important for the quality of life, medical systems, for example, as well as for the environment. And what's interesting is that making a molecule is a somewhat challenging effort because you need to bring in subunits to make a larger molecule. And then you have to start wondering, well, particularly in the material side of applications, how do they come together? And how does that collection of chromophores impact a particular technology that you're trying to optimize? And the stories that I'm going to tell you are associated with those ideas, and also, perhaps more importantly, the people who did the work in the lab and how they were able to bring that knowledge into the real world. So one of the molecules that we made, or one class of molecules that we made, is shown here. The details are perhaps not so critical, but I'll go over them a little bit. So it's a conjugated molecule where we have an electronically delocalized structure that we bring electron-rich and electron-poor units in a particular sequence. And it turns out that those molecules have the optical and electronic properties that you might want to use in the fabrication of a solar cell. Now, when you make a molecule and you're trying to sort of think about, well, we're going to make a film, a thin film, or an object out of it, it's important to understand how they're going to come together. And for that, we use techniques such as grazing incidence, wide-angle X-ray scattering, which allows you to understand the local order within molecules in a very thin film, say 100 nanometers thick. And by looking at the diffraction patterns that came out of those thin films, we were able to understand the organization of the molecules in the thin film and correlate them to crystal structures from which we knew the distance, the orientations of the molecules relative to each other, and from which we could calculate electronic coupling, carrier mobility, conductivity, etc. And with that package of information in hand, we started thinking about making organic solar cells. So here's one of the molecules that I made. Here's another one. We mix them together. They create a blend, almost like an oil-water mixture. Light comes in. It creates an excited state, or an exciton in the language of physics, which finds itself onto an interface where there's a driving force for electron-hole separation. And if the object is organized just in the right way, you can collect the electrons and the holes on the electrode, from the electrodes, from which you get a solar cell. And again, everything sort of stems from the fact that we can design and optimize the molecular subunits that go into that solar cell. Now, what's interesting is that we had this, it was a new class of molecules. These small molecules had never been used before. We knew how to process them. And the student that was principally in the group Corey Hogan, shown here, together with other members of the teams, such as Tamayo and Andres and Bruno, decided to take advantage of that intellectual property and form this company called Next in Santa Barbara, California. And the idea there is, unlike silicon solar cells, organic solar cells allow you to make transparent solar cells. And if you see an image of the Singapore skyline, there's a lot more windows than rooftops. And so the idea there is that the windows could be turned into solar cells. And in fact, last year, the Next Energies got awarded as the most disruptive technology by the venture capital funds in the central coast of California. So again, the idea of thinking about a molecule, how it comes together, how we bring many molecules together, how do we organize the organization, how we control optical electronic properties, very useful in terms of being able to do this subsequent application. I'm going to give you a different story now. Again, this is sort of like a train ride kind of stories, and it's associated with a work that came from the lab that enabled the design of the most widely used antibody products, antibody dye products. And it's built on a pi conjugated backbone, these conjugated polyelectrolytes. Here's one specific example. Here's the backbone. There are some subunits from which we pick the color, the ionization potential, the electron affinity. So we can tailor the backbone. We can tailor the distance between the charges on the backbone. We can pick what kind of cationic charge you want, one pyridinium, tetraalkyl ammonium, okay, up to you. We can choose the counter anion, or we can do charge reversal. We can make the cationic, the polymer anionic instead of cationic. And if you think about a polymer structure, particularly a conjugated polymer structure, if N here, the number of repeat units is very large, they're not likely all to be coplanar with each other. Entropy will tell you that there's some structural deviation, twist, there may be a structural defect. So you can think about them as a collection of chromophores attached to each other. And unlike a collection of chromophores or dyes, if you want, in solution, there is an electronic communication between these subunits. So if this particular fragment absorbs light, then there's the mechanism by which that initial excitation can travel to a lower energy site and find its way there. And you'll see why that's interesting in a minute. The mechanism by which that process takes place is described by the Forster equation. So you can think about it in terms of a probability, but the probability, of course, increases if the rate of energy transfer is increased. And the rate of energy transfer is shown here, which is a set of fundamental constants. And these four variables, n is the index of refraction of the medium. We don't have any choice, but the distance is important. It varies 1 over r to the 6th. And there's these other two parameters. J is the overlap integral, which tells you the overlap of the emission of the donor and the absorbance of the acceptor. And kappa is a tough term to really come to terms with, but it tells you the orientation between the transition moments of the donor and the acceptor. Now, that's a mouthful, but the important thing is that we have these parameters onto which we want to map specific molecular molecular structures. We also need to take into account where are the absolute energy levels of the donor and the acceptor. If the acceptor energy levels is contained in that gap, then we get energy transfer. And if it's offset, we get charge transfer. Now, this process is the one that leads to the essential charge generation in solar cells. This other process allows us to move excitations along the polymer. So what can we do with this? How can we organize all of these elements into something useful? Well, let's start with a simple idea where we have a conjugated polymer, polyelectrolyte that's cationic, and we have a PNA with a chromophore. Now, everybody knows the structure of DNA, the sugar phosphate backbone, and the nucleosides responsible for recognition. A peptide nucleic acid simply replaces that sugar phosphate backbone by a neutral peptomimetic. So this is neutral, okay? And then we can add but there are no electrostatic interactions, and that's the key point. we can add DNA and we can ask ourselves if it's complementary or not to the PNA sequence. And if it's not, because the DNA is negatively charged, it will make an electrostatic complex with the conjugated polyelectrolyte, and okay, that's the end of that. But if the PNA and the DNA are complementary, then all of a sudden, that dye that you put in the PNA goes close to the polymer, you excite the polymer with light, and you get energy transfer to the dye. Very cool, very cool. And you can see that allows us to determine whether there's a matching DNA in solution, and more importantly, the signal intensity if you go through the energy transfer process relative to the direct excitation is greater by a factor of 100. So that was the basis. Again, I know I went fast, but just bear with me or accept with me that we can do these optical amplification process through electrostatic interactions. Let's go back to molecular design. This was actually one of our breakthrough polymers where we have introduced a small fragment of these benzothiadiazole units into a polymer backbone, which is rigid. It's phenylene, fluorine fragments, but the transition moment of this fragment is orthogonal to the transition moment of the backbone, so there's no energy transfer. And because of that, we can actually do cool things. And now we go back to sort of more the application, easier to understand essential elements of function. So let's look at this. Let's start again with the chemical structure that I just showed you. Here's the benzothiadiazole. The polymer typically emits blue. There's no energy transfer along the chain. If now we add, again, the PNA, the peptidonucleic acid here in orange, here's our DNA. If the PNA and the DNA, single-stranded DNA, don't hybridize, then you make this electrostatic complex that then forms a larger order aggregate, these sort of coacervates. And in this coacervate, all of a sudden, the transition moment is now not strictly defined, and we can get energy transfer, so the solution turns green. If, however, we do get hybridization, we make a complex to the PNA that bears this dye, and instead of glowing green, it glows red. So that's the very important thing, and here are the actual data that demonstrate that. So at the time of this work, maybe 15 years ago, nobody had been able to demonstrate that you could get optical amplification through energy transfer of a conjugated polyelectrolyte. And at that time, in conjunction with my student at the time, Brent Gaylord, and a colleague of his and now a collaborator of mine, Patrick Dickinson, we started a company called Sirigen in 2004 to develop this into something actual practically useful. And around 2012, I'm sorry, I don't know why the date is not here. It was actually purchased by Becton Dickinson. Becton Dickinson is a large supplier of chemical reagents and biological reagents because we had shown that they're very effective at labeling antibodies used in flow cell cytometry. So now one of the top choices for using labeling cells and antibodies in flow cell cytometry are based on these dyes that we originally developed at Sirigen. And everything else, and there's even an app for it. I mean, they sell an app. I never knew that you could make an app out of new molecules, but they did. I thought that was really special. And here's, whenever there's a commercial opportunity, there's another company that wants to get in. So about a year and a half ago, 15 years later, there was some litigation, and we had to go and show the notebooks that were used for examples in the key patents, and this whole affair was settled in court. And now, what's interesting is that Becton Dickinson shows all the papers that use their reagents, and all of these papers came out in 2018 that used the reagents we developed in Sirigen. Here's another. And if you look carefully, you can just pick random papers on stem cell, on immunology, on clinical development, on aging, on microbiology, on cancer. So this whole idea that you can start with a molecule that you pre-program function that's unique with a view of a critical application, and is not only do you get nice papers, not only do the people that work in the project do very well later, either if they want to go into academia or in business, and but it makes impact in society more broadly speaking. Just a couple of other, just maybe two more slides. I think I have a little bit of time. Um, Another story about students in the group, and you know, we've heard from Professor Rong Li regarding the importance of interdisciplinary research, and this will bear some interesting influence. So these are 5 students from the group. At the time, Jenny worked in microbiology. We were looking at transmembrane electron transfer. James and Lou were working at the solar cell project that you saw there. Uh, Ron is a hardcore chemist. Ron is the kind of guy that can, if you can draw the molecule, he'll make it. And Thomas was involved in understanding the photophysics of these molecules. And they basically graduated, left the group 2012. They started a company called Appeal Sciences. And what's interesting is that diversity of expertise and ability to work together across many disciplines enabled the creation of this company, which essentially makes edible coatings that keep fruit fresh for longer periods of time. And now the current valuation is about $1 billion. The largest investor, strangely enough, I didn't know that, is the Singapore government, Oprah Winfrey, people like that. And it's now sold throughout the US and Europe. Here is what it looks like now. The company, I'm super happy to see our ability to train people to do science well and critically and work together to do something later on in society. Here's my thank you slide. But before I say thanks, I want to tell everybody out in the audience, you know, I came to Singapore in January of this year from California. And why? Well, I'll tell you why. I was very, very impressed with NUS. And not only the diversity of people, cultures, academic endeavors, entrepreneurial activity, fantastic, really fantastic. There is deep expertise in any area of academic research and scholarship. So whatever your choice may be, guaranteed you'll be served by the very best in the world. There's excellent support for entrepreneurial activities, and there's very bold university leadership that really values the success of its graduates as a metric by which they consider their university or their objectives to be successful. So you are coming to the right place. And you will be welcome, whether you're from India or China or US, Canada, Europe, Southeast Asia, Japan, Africa, South America, Australia. Singapore is a very welcoming society, multicultural. You will be made feel at home without any worries. So if that's a concern, I assure you, it's not going to be a concern. So I think that's what I had to say. Thank you for your attention. I'm happy to answer any questions that you may have.
Thank you very much, Professor Bazan. I think very exciting research and very kind words about Singapore. Okay, so we are now open for questions from the floor. Maybe I'll start with a question. You have very exciting technology that you have used in many applications and grew companies. Have this technology been used to identify our current COVID virus?
Well,
Or is it possible to do it?
Well, no, I'm glad you asked. And I have some backup slides. So we came here around February or so.
Mm-hmm.
There was a lockdown, as discussed before. Labs weren't available, but we were able to work in collaboration with colleagues and develop in applying the work that we did in the conjugated polyelectrolytes to improve or lower the cost of RT-PCR. So I think I have a couple of things. So if the way that this works is you take the viral particle, you extract the RNA. This is just like any normal PCR assay, but the way that we're approaching it is that in the PCR amplification step, we actually label some of the nucleosides with in this case fluorescein, which is an optical reporter. It gets incorporated into the DNA, and then we add the conjugated polymer, the conjugated polymer. We excite the polymer, and then we get energy transfer to those dyes that have been incorporated during the PCR reaction. And it works very well. In fact, you can see some interesting information here. So you can see with and without the dye, sorry, with or without the original RNA, we get a very clear signature. You can even see it by eye. This is no COVID RNA with COVID RNA. And it's not, I think, super revolutionary, but we think we can certainly lower the cost of every single PCR assay by, maybe $10. And when you do as many PCR protocols as are being done around the world, this might have an opportunity to be successful.
Wow, interesting. Any questions from the floor? Maybe the second question maybe I'd like to ask is that many of your students and postdocs become CTOs and CEOs. Maybe you can share with us a little bit about what makes which kind of students will make good CEOs or CTOs? And yeah.
I don't know, but I will tell you one thing is that for sure, everybody that became a successful CEO or CTO was very good at the area that they chose to study in graduate school. They all published high quality papers. They all were able to have domain expertise in the subject that they chose to study. And at the same time, I would say that they were able to communicate with people. They were interested in science beyond the area that they're studying. They were very good at interdisciplinary and multidisciplinary research, and they were pretty balanced. So, for example, Corey, who's CTO of Next, he came in with both anthropology and a material science degree. Brent, who was the CTO of Sirigen, he was actually a graduate from the Air Force Academy. So he was very good at a variety of leadership skills, and he was a very, very good material scientist. Jenny Du, who is the operations manager at Appeal, she was always the kind of person that she could get people to come and work with her. She was very sort of persuasive in terms of getting people to rally around her cause. And James Rogers, who is now actually Forbes magazine, the business magazine, said, put him on the 40 top businessmen under 40 years old.
Wow.
In the United States. Well, he was also same qualities as Jenny, but also, he was very balanced at the same time.
So what kind of advice would you give to people considering doing PhD? Is it worthwhile if they are thinking of being a CTO or CEOs?
Well, first of all, you need to master the one thing. I mean, everybody says about that interdisciplinary research is important, and it is very important, but you need to integrate yourself. You go to a table where the people are going to discuss some interdisciplinary project, but you need to be the best at what you decided to bring to the table. So they were very good at that. Pay attention about your presentations, pay attention about how you communicate with people, and have the bigger picture. You know, I would say that those are qualities that anybody can improve on. No, you know, everybody, we can all get better all the time. So I would encourage people to be effective communicators, to make, you know, when you talk, you want people to understand. You don't want people to work hard at understanding. And so all of those people were very good at that kind of skill. And many of them developed it in time. And they weren't necessarily born with it, they just worked at it.
Okay, there's a question from Sean Tang who says, do graduate students have the opportunity to work with patents?
With patents?
Yes.
Yeah, very good question. You know, yes. If your work comes out as innovative and you want to draft a patent, the process is you give the university a disclosure, and then the university will hire a patent attorney to actually draft a patent with you. And you will find that process very interesting. The whole idea of integrating an invention into a legal document and the strategies that you develop to protect that idea is a totally different, very fascinating intellectual enterprise. So yeah, if you do that, if a patent or innovation from your research comes out, for sure you're gonna do it, for sure.
Okay, thank you very much, Professor Bazan. It now gives me pleasure to